My name is Nigel Rogash and I'm a Senior Research Fellow in the Adelaide Medical School here at the University of Adelaide. I'm also your MC for tonight. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains and the lands on which the University of Adelaide campus at North Terrace, Waite and Roseworthy are built. In tonight's presentation, we turn our attention to in vitro fertilisation, or IVF. As many of you know, IVF is widely used across Australia and the world to help parents in waiting start a family. Currently, 1 in 20 Australian births are IVF babies. However, with the success rate per embryo still down around 25 to 30%, there remains much room for improvement. Tonight, you will hear about one innovative approach. Our presenter, Dr. Kylie Dunning, and her team are developing revolutionary new light-based technology to measure embryos' likely success when transferred back into patients' wombs. Kylie will discuss her research and share how it could help people become parents sooner. Following her presentation, I will open up the Q&A session with our online audience. We encourage you to submit questions during the presentation via the Q&A section down the bottom of your screen. I'd now like to welcome Dr. Kylie Dunning to the stage. She is a Hospital Research Foundation Fellow in the University of Adelaide's School of Biomedicine. Kylie leads the reproductive success team within the university's Robinson Research Institute and ARC Centre for Excellence for Nanoscale Biophotonics, and was also the 2020 SA Young Tall Poppy Science Award winner. Welcome, Kylie.
Thank you, Nigel, for that kind introduction, and thank you for the invitation and the opportunity to speak to you all this evening about some of the work that we do in our group. So I'm a parent to this gorgeous little girl and to these handsome little fellows too, and I'm extremely fortunate and privileged to have this role in their lives. I definitely have a lot fewer hours sleep under my belt and many more wrinkles since having them, but I wouldn't change that for anything. However, like many others, my journey to parenthood was not smooth sailing. And to describe it as an emotional roller coaster is a gross understatement. But it's this life experience that motivates me every day at work. I'm really passionate about helping parents start a family. So in our group, we focused on the very first few days of life, so the first approximately 5 days, and that are events that occur post-fertilisation. So you can see this process happening in the reproductive tract, depicted in the reproductive tract here. This is when the embryo goes through a series of rapid cell divisions. And I have a nice time-lapse video here that shows this nicely. So here you see a single cell embryo that has undergone fertilisation. And then within 24 hours, it divides into 2 cells, and then 4 cells, 8, and then 16. And then development continues until it reaches this stage of development. And this is termed the blastocyst stage of development. And this is the first stage in development when 2 distinct cell populations arise in the embryo. And these cell lines are very important for subsequent development. So this embryo is made up of a group of cells called the trophectoderm, and these go on to form the placenta and then all the other extra embryonic tissues. And then the inner cell mass, which goes on to form the conceptus and eventually to form the fetus. And I want to, I'm introducing this term now because I continue to use this terminology throughout the presentation. So it's estimated that 1 in 6 couples in Australia will have trouble conceiving. And globally, this equates to over 48 million couples every year. And for most, the treatment for infertility, for most of these people, they turn to assisted reproductive technologies, which includes IVF. I just thought I'd briefly, for those of you in the audience not familiar with an IVF cycle, I thought I'd just briefly take you through the overall steps involved. So the oocytes or eggs are harvested from the woman following large doses of hormones to stimulate the growth and development of multiple oocytes. And sperm is also harvested at this point. Then the process of fertilisation happens either by co-incubating the sperm, shown here in blue around the outside of the oocyte, or through direct injection of a single sperm into the oocyte. And then the embryo is allowed to develop in the lab, just like in the time-lapse video I showed you earlier, through a series of cell divisions until it reaches this blastocyst stage of development. And this stage is also important because it's the point at which the embryo can be either transferred, shown here, back into the uterus or womb to implant and establish a pregnancy, or embryos can be frozen, so cryopreserved in liquid nitrogen for use later. So IVF has come quite a way since the first baby, Louise Brown, was born more than 40 years ago now. We've certainly come a long way. I mean, we do not grow embryos in the lab in this glass dish as Louise Brown's embryo was. We've also gotten a lot better at maintaining temperature. We've got better at growing the embryos in a nutrient growth medium that tries to best replicate that in which it grows inside the uterus, and other things as well, like maintaining pH. And certainly more and more people are seeking IVF. So this graph here depicts the number of IVF cycles shown on the y-axis since 2002 to 2007. And what we see is a doubling of IVF cycles initiated over that period. But if we look at the number of cycles that resulted in a live birth, these have really remained stagnant for the last 10 years, with approximately 18% of initiated IVF cycles resulting in a live birth. So in our group, we're interested in developing novel technologies to determine in a non-invasive way which embryos are the healthiest in order to hopefully result in a pregnancy and a healthy baby. So some of the current tools that are used in an IVF clinic to determine which embryo should go back first are things like point-in-time observations. So these are really the embryologist taking the embryos out of the incubator, so the warm environment in which the embryos are grown, and looking at whether they've met a specific developmental stage within a particular time. They've also, many clinics also use time-lapse imaging, so just like I showed you very early on in the presentation, so whether embryos, at what point, how many hours post-fertilisation they're reaching each of those developmental stages, whether they be 2-cell, 4-cell, 8-cell blastocyst. The overall limitation in doing this is that these measures are not associated with live birth outcomes, so they're not overly accurate at predicting whether an embryo will result in a live birth following transfer. Other people have looked at measuring metabolism. So metabolism is how cells generate energy from building blocks to do many processes like cell division and developing into different cell types. We also think this is a very important in embryo development, and we think it's probably a very likely promising tool in predicting embryo quality. But the current means in which people have, ways in which people have attempted to do this so far is to do things like take the spent medium. So this is the growth medium that the embryos have grown in in the dish in the lab over a say 5-day period. Others have taken different approaches, like putting an oxygen probe close to the oocyte or the embryo, and because oxygen consumption is required for metabolism as well. But some of the limitations involved in this process is that they are both point-in-time observations, so you're really getting a snapshot with spent medium analysis really at the end of development. And you're also getting a picture of the whole embryo. So you really don't know what's going on in individual cells of the embryo or in different cell types. So say the difference between the inner cell mass, which forms the fetus, versus the cells that form the placenta. So we think that we need new methods to evaluate embryo health. So we're motivated to develop accurate, objective and non-invasive methods to measure embryo health. And for this, we have turned to light. So light is used in many different areas to take measurements. Light was used to measure gravitational waves. It's used in the hospital. So when you, it's something called a pulse oximeter. So everybody would probably know someone or themselves have had that little clamp put on their finger. So that uses light to measure oxygen levels in your blood. And we're really interested in understanding whether we can use light and gentle doses of light to measure embryo health. So we use different wavelengths of light. So that's just shown in a picture here, like different wavelengths, different colours of light, because we know that different colours of light will actually excite or make glow different molecules found within cells. And of interest here are two types of molecules. One is called NAD and the other ones are called flavins. So, like I said, we think metabolism is an important determinant of embryo health. And this picture here is, it's very complicated, so don't worry too much. This depicts a process that happens in mitochondria. So these are the powerhouses inside every cell. And these mitochondria make energy in the form of ATP. And the important thing to know here is that two of the molecules used in this biological process are distinctly autofluorescent. So if we shine a particular wavelength of light on a cell, the molecules that glow back provide us with a measurement of what's going on with these particular molecules, and we can use it as an indicator of metabolism. So the approach that we've been using to use light to measure embryo health is something called hyperspectral imaging. So this type of imaging or microscopy can capture thousands of spectra using multiple excitation and emission wavelengths. And the other great thing about this approach compared to some other types of imaging available is that it uses very low levels of light on the embryo and a very powerful camera to pick up the low levels of light that glows back from the embryo. So interestingly, hyperspectral imaging has been around for a while and used in several other processes. And I've got a couple of interesting examples here to show you. So on the left here, you can see these are actually frozen potato chips. So they're used in food quality analysis before batches of food go out. So here they're looking at whether their batch of frozen chips is good enough to hit your supermarket shelf. To look at the integrity of chicken eggs, and also whether your kiwi fruit is perfectly intact with no bruises before you pick it up from the fruit and veg shop. It's also been used in art history. So this example here is actually Leonardo da Vinci's Virgin on the Rocks painting, and hyperspectral imaging was used to show the original drawings that are found beneath the final painting. More recently, this type of imaging has been used in cell biology. And in this example I show here, the imaging was able to discriminate between cancer cells with low and high survival rate. And this is really amazing because molecularly, like on the genetic level, these cells differ very little, but the imaging was able to pick up differences between cancer cells with low survival and high survival. And the other thing I wanted to point out here is that this type of imaging gives you an indication of what's going on between cells. So you can see in this case down here, you can see this one is false coloured with a low level, whereas this cell here has a high level of the fluorophore or the molecule that's being assayed. And you can also see differences within cells as well, so where particular molecules are found within cells. So hyperspectral imaging provides an alternative approach to measure metabolism within and between individual cells in a non-invasive way. So we are interested in understanding whether we can use hyperspectral imaging to detect a particular process that can happen during the early days of embryo development. So like I played this earlier, this embryo undergoes a series of very rapid cell divisions, and within 5 days it's gone from one single cell type to 2 different cell types. And the next 2 slides give a really nice overview of what can happen when things don't go to plan. So in this example here, this is a cell, it's not from an embryo. The chromosomes, so that's the genetic material, the chromosomes are stained in green and the rest of the cell is stained in red. So during normal cell division, the chromosomes will be doubled, and then as the cell divides from 1 into 2, there will be equal segregation of chromosomes into both cells. And so I'll play that for you now. So the chromosomes align at the centre in what's called the metaphase plate, and then you get equal division of chromosomes into the 2 daughter cells. And that is termed euploidy. But when it doesn't go to plan, sometimes, oops, I think we need the next slide, this one. But when it doesn't go to plan, sometimes a chromosome gets left behind. You can see this one here hanging out in the centre. So this chromosome ended up in the bottom cell when it should have ended up in the top cell. So what we have is one cell with too few and one cell with too many chromosomes, and this is termed aneuploidy. So aneuploidy can arise in the early embryo. In fact, many human embryos are found to be mosaic, meaning they contain both euploid and aneuploid cells. And it's this type of mosaicism that's predicted to cause early pregnancy loss in both IVF and naturally conceived cycles. But we do know that some human mosaic embryos do lead to a pregnancy. But from studies in mouse models, we know that a high proportion of aneuploid cells in the embryo do not go on to form a successful pregnancy. So it's almost like there's this balance, like that there is perhaps a level of aneuploid cells that's tolerated and you'll get a pregnancy and a healthy baby, and perhaps if the level is too high, the embryo won't implant and a pregnancy won't establish. So to assess aneuploidy and mosaicism in the clinic currently, an embryologist will take a biopsy of cells from that day 5 embryo, from that blastocyst embryo. So you can see this process here. So you see 5 to 6 cells being sucked up into the pipette, and this thing here is a laser that's being used to cut the junctions between cells in order that biopsy is torn free from the embryo. And then that biopsy of cells is then sent off to be sequenced to give us a picture of how many chromosomes are present in each cell. So whether the biopsy sample is euploid, aneuploid, or whether there's some level of mosaicism, so some euploid and aneuploid cells. At this point in time, it's good that I should probably say that there is evidence to show that this procedure is associated with an increased risk of preeclampsia if an embryo has been biopsied and then transferred back to the uterus. There is evidence that there is an increased risk of developing preeclampsia, and this is a pathology of pregnancy that you don't really want. It can lead to high blood pressure and the need to deliver a baby to resolve it. That also predisposes the mother and the baby to an increased risk of cardiovascular disease later in life. The other thing that's becoming pretty clear from this process is that the biopsy taken isn't always reflective of what's going on in either the rest of the trophectoderm cells or the cells that go on to form the fetus. So I did forget to mention that the biopsy is taken from the trophectoderm. So those are the cells that go on to form the placenta. So I think that this example here, in which a study showed that if you take a biopsy of cells from the trophectoderm, so say you took the biopsy from this quadrant of the trophectoderm cells, the placental cells, and you sequenced it, that would give you a correct representation of what's actually going on inside the cells that go on to form the fetus. However, if the biopsy was taken from this quadrant of the embryo, the placental cells, it would have been diagnosed as mosaic. So 1 in 3 cells were aneuploid, when in fact the inner cell mass, the fetal cells, were genetically normal. So cell biopsy of the trophectoderm does not give an accurate indication of the proportion of aneuploid cells in the inner cell mass in all cases. And the best way to depict in which cases it's accurate and which cases it may not give you an accurate representation is shown here. So in these pictures, the aneuploid cells are shown in red and the euploid, so expected number of chromosomes, are shown in blue. So in this case, the inner cell mass is mosaic. So the possible biopsy is one in 3 chances. It would have got diagnosed as aneuploid, euploid, or mosaic. So there was only a 1 in 3 chance that this embryo would have got a correct diagnosis. In this type of embryo, the biopsy is not an indication of what's going on in the fetal cells. This one here, same, 1 in 3 chance of correct indication. And these final 2 scenarios, the trophectoderm biopsy would not give you a picture of what's going on inside the fetal cells. So the current diagnostic method fails to provide information on mosaicism type or the proportion of aneuploid to euploid cells. So like I was saying earlier, perhaps some aneuploid cells is okay and tolerated, but perhaps there's a tipping point. So if we could get a picture of the level of aneuploidy, it might be a good indicator of whether that embryo would result in pregnancy and live birth. So we are really passionate about seeing if we can find a way to create a map of how many aneuploid cells are present in the embryo and where exactly are they. So we already know from published studies that human embryos that are aneuploid have altered cell metabolism. So if embryos containing aneuploid cells display altered metabolism, can we detect this non-invasively using hyperspectral imaging? To answer this research question, we've taken a couple of different approaches, a couple of different models. The first model we've used are fibroblast cells. So these are cells that are skin cells that are they're human skin cells. And these are characterised as being either euploid or aneuploid. And then we've gone and imaged these cells and then used a series of mathematical algorithms on top of these images to determine whether we can use this approach to say, yes, that cell is aneuploid and that one is euploid. So because we're interested in whether at first change aneuploidy leads to altered metabolism, so can we confirm what's previously reported in the literature? We can use this hyperspectral imaging to quantify the abundance of those 2 molecules that are used in metabolism that I said are important in generating energy in a cell. So we're able to first get a heat map of exactly where these molecules are within the cells, and then use that over many, many cells in different experiments to quantify the abundance. And we find for NADPH that aneuploid cells have significantly higher levels of this molecule. In terms of FAD, so this is the other molecule that's used in energy production, we find that aneuploid human fibroblast cells have significantly lower levels of FAD. And we can take these quantified abundances of these 2 molecules and use this well-used equation it's called the optical redox ratio, but what it really gives us is a picture of the overall metabolism going on inside cells. And so using this, we see that aneuploid human fibroblast cells are performing significantly lower levels of energy compared to euploid cells that have the expected number of chromosomes. And then, like I said, we have our collaborators have access to quite a few, 33,000 feature algorithms. So these are mathematical code that are put on top of the autofluorescence that we have taken using the hyperspectral microscopy. And then using these algorithms, the goal is can we use that to say that cell is euploid and that cell is aneuploid? And that's shown nicely here in this plot. So using two of the features here, we can see that the euploid cells separate quite nicely from the aneuploid cells shown in red. And to test whether this approach is accurate, we can plot something called an ROC curve. And the important thing about these curves are you want to be as close as you can to the top left-hand side. So that means high level of high positive rate and very low false positive rate. And for this approach, we have a very high accuracy of 0.97 when we look at the area under the curve. And this is great, right? So this was a great first step. We should show that this approach works in human cells from the skin that we know are euploid or aneuploid. But of course, we want to know, does this work in the embryo? So to do this, we first looked at individual cells from the embryo. And for this purpose, we are using the mouse as a model. Because mouse embryos have a naturally very low level of aneuploidy, we use a chemical that's called reversine that temporarily affects the cell division process, and we use this to induce aneuploidy in the mouse embryo. And we do this during the embryo's transition from the 4-cell stage to the 8-cell stage. At this point in time, we dissociate, so we pull apart all the different cells. And then I'll just take you right through the whole process. In order to get blastocyst embryos with different levels of euploid and aneuploid cells, we then reaggregate these cells back together in differing ratios. And these amazingly develop to the blastocyst stage to develop embryos with varying levels of euploid to aneuploid mosaicism. So like I said, we first looked at this point in development. So could we detect differences between these individual cells from the 8-cell embryo? And similarly to the human fibroblast cells, we can see higher levels of the NADPH molecule in aneuploid cells and lower levels of FAD. And this was again reflected when we quantified this and plotted it on the graph. So this pattern exactly matches what happens in the human fibroblasts. We again confirm that the overall optical redox ratio, so the overall level of metabolism, is significantly lower in aneuploid cells from a mouse embryo. When we apply the features analysis on top of these cells, we get very, very nice separation of these two cell types. And this is reflected in an extremely high area under the curve. So a high positive, low false positive rate. But of course, the penultimate question we want to know is, can we do the same specifically in the inner cell mass of the blastocyst embryos? So can we determine the level of aneuploid cells in the cells that go on to form the fetus? So for this approach, we just looked at the fluorescence that came from the inner cell mass. And while we saw the same pattern, so in NADPH, we saw higher levels of NADPH in aneuploid embryos, and lower levels of FAD or flavins, and an overall lower level of metabolism. This didn't reach statistical significance. However, we still went on and applied our features analysis directly on top of the autofluorescence, not on the quantified abundance of these molecules. And we still got quite good separation using this approach, with an area under the curve of 0.87. So we think this is a great start. We were also interested in challenging the process a bit harder. So could we really distinguish between two embryos that had closer levels of aneuploid cells? So embryos that were a 1-to-1 ratio versus a 1-to-3 ratio. And we didn't see a significant difference in NADPH, but we did see significantly lower levels of flavins, or FAD, and significantly lower levels of metabolism overall in embryos that had higher numbers of aneuploid cells. And again, the features analysis on top of the autofluorescence shows great promise with an area under the curve of 0.88 using this approach. I guess the thing that we are also very cognizant of is that whatever approach we take, we want to ensure that this is not damaging the embryo. So in most other situations, people aren't really worried about if they image a set of cells or a cell that comes from a biopsy and then is thrown away. People aren't concerned with whether that light is inducing any damage itself. So we think it's very important to assess the impact of any new imaging technologies on embryo health, and we've done a bit of that work on Hyperspectral. So we've taken some morula stage embryos. So this is the stage before blastocyst stage, and then embryos were either imaged or not imaged, and then we saw whether they were capable of developing to the blastocyst stage and whether the level of DNA damage differed between embryos that were imaged or not imaged. And we found that there was no difference in embryos reaching that final stage of development in vitro, and the levels of DNA damage were comparable and not significantly different between non-imaged and imaged embryos. But of course, the big question is, do the embryos that were imaged go on and form a live offspring? So we took embryos that were either imaged or not imaged, and then they were transferred to pseudo-pregnant mice. And we can see here some healthy live pups born from mouse embryos that were imaged. And we also looked at the weight at weaning, so this is a good indicator of health when they are ready to leave their mother. And we saw no difference in the pup weights between pups that were derived from embryos that were not imaged versus imaged. And we have the final step that we are keen to do is to confirm that the results we are seeing are definitely due to aneuploiding. So we have taken embryos and specifically isolated the inner cell mass. So there is a series of pictures to show here. So this is a blastocyst embryo being held under suction by a holding pipette. And this is the group of cells here you can see are the inner cell mass. So Ryan Rose, who's a very highly skilled embryologist, has helped us with this to specifically take the inner cell mass, which is located now in this pipette, with the remaining trophectoderm cells still over here on the holding pipette. So we're going to confirm that all our embryos are in fact euploid or aneuploid. So we think that hyperspectral imaging is a promising tool for determining embryo health. But like this turtle here found on this fence post, while I'm the person who was invited to speak here tonight, I did not get here like this turtle on my own. A colleague of mine uses this picture, and I think it perfectly depicts how I feel about this project and the group of people that I work with. So this project really came about because of my introduction to cross-disciplinary science in the Centre for Nanoscale Biophotonics. So it was really, me, the biologist, being exposed to a physicist. And no, it was not like Amy and Sheldon, but it was a great group of people. It is a great group of people to work with. And here you can see examples of us building teams together to develop new science. And I'd like to finish by highlighting some of my team here. All of the team is fantastic in the reproductive success team. I'm very fortunate to have a great group of young researchers work with me. And I'd also like to thank our collaborators at UNSW who have done all of the features analysis of the algorithms. And I can't stop by thanking the sources of funding who let us do what we do every day. We are fortunate to have this job and love this job. So what does that all mean? Well, we're here because we want to provide hope. Hope to families to start a family. I hope to parents to start a family. Thank you.
Okay, thank you, Kylie. That was a fantastic presentation and very, very well done under the circumstances as well. Okay, so just a reminder to those of you that are listening along at home, if you have a question you would like to ask to Kylie, please type it into the Q&A section at the bottom of your screen, and we'll do our best to get to those. So, while some Q&A questions are coming through, we have a couple of pre-submitted questions. So the first one is, when will this technology be used in Australian IVF clinics?
That is a good question. I think we've got a little way to go before that happens, including verifying that this approach is safe in other models before we introduce it in the IVF clinic. But yeah, I'm excited to continue to work in this space and to develop novel technologies that we really want to translate into useful techniques for the clinic.
Great, and here's another pre-submitted question. How can light technology be more accurate than a biopsy?
Yeah, that's a good question. So if you remember, I showed a series of different, I used diagrams to depict the different types of, and like where aneuploidy could be found in an embryo. So if you take a biopsy from a set of cells, the cells that go on to form the placenta, there is no guarantee that biopsy is reflective of what's either going on in the rest of the placental cells or what's going on inside the cells that go on to form the fetus. So I didn't actually say that, so in the placental cells, aneuploidy is quite well tolerated. So the placenta will, under most circumstances, will implant and function quite well, even if it contains aneuploid cells. But in the fetal cells, there are some types of aneuploidy. Most aneuploid types are not compatible with life. So that's why using light to really look at what's going on inside the cells that form the fetus is likely to be a more accurate approach than taking a biopsy from a totally different cell type.
Okay, great. And another question. So practically, what are these light cameras like? Are they something that could be used in clinics, or do you need a special set of equipment to actually run these kind of analyses?
Yeah, so I guess well, at the moment we assess the whole. We take pictures across the whole spectrum, so a wide, lots of different wavelengths, right? So. But it's likely that there'll be a few wavelengths and a few molecules that will be important in determining embryo health. So by the time something goes from a microscope and a camera that sits in our lab that's large and clunky, by the time it gets to the clinic, this will be a much more refined device that is user-friendly. You won't need a physicist to run all the algorithms.
Okay, great. So I can't see any further questions coming through. So I'd like to finish off now by thanking tonight's speaker, Kylie. You delivered a really fantastic and engaging presentation. Thank you very much. And for those of you who were able to stick around, I'd like to thank you, our audience, for viewing and for your questions and comments tonight. We look forward to inviting you back in September for our 3-minute thesis final. Now, the 3-minute thesis is a highly popular event because it provides an opportunity for our audience to hear from 10 of our top PhD students. Be sure to sign up for our mailing list to receive the latest information and of Research Tuesday and all the related news. So thank you again for tuning in, and again, apologies for all of the technical delays tonight. We hope to see you again next time. Good night.